And now it's time for our last session of this conference, the time for roundtable discussion. For those who joined us recently, my name is Vikram Devgan. I'm a VP of Global Marketing at Bruker Cellular Analysis and going to be your host for this discussion. With me, we have Dr. Anthony Zamora, Dr. Stefan Eichmüller, Dr. François Romagné, Dr. Xiaoxiao Wang, and Dr. Shu Wang. First of all, awesome talk, everyone. Very impressive. A lot of good work is going on in your lab with Beacon platform. So it's it's always good to see how the Beacon platform is advancing research. And we did get a lot of questions from audience during this conference. So I'm going to use few of them to start this session. So let's start with questions for cell therapy. And these are for Anthony and Stefan. The first question is, there are multiple technologies for CAR and TCR discoveries. Can you share your views on effectiveness and complementarity of leading technologies? So maybe I'll request Anthony to start.
Yeah, sure. Happy to help answer that question. So I think, the technologies kind of differ depending on whether you're looking to, discover and also validate TCR T-cell based therapies or CAR T-cell based therapies. I think some of the initial, technologies and assays that are typically performed are more looking at, the binding affinity of a specific CAR construct for the antigen. But really, you get the most information from, the functional studies. So these could be, various cytokine secretion assays, all the way from ELISAs all the way up to, multiplex single cell assays using the IsoLight or IsoSpark. A lot of them are focused on cytotoxicity. So there's various instrumentation like Excelligence or InQSight, where you can culture CAR T cells with their targets and measure the extent of killing those tumor targets as an indication of how functional specific CAR constructs would be. And so these assays are effective at giving you kind of a global view of how CAR T cells or TCR T cells, would respond to specific antigens, but they really give you less insight into other important biological parameters, right? So you're typically, characterizing a single parameter and then extrapolating across different assays. And so really, I'd say that they could be complementary in terms of, they're a good starting point if you want to do bulk and get a general sense of how well a specific, CAR T cell or TCR T cell are going to function. But really, it requires single cell resolution to really understand the heterogeneity of the response. And then also multiplexing, the different parameters. So looking at phenotype, looking at cytokine secretion, looking at cytotoxicity really gives you strong correlation with what you would expect to see once these therapeutics are infused into a patient. So some of them are complementary, and then some of them I would say are good measures for an initial indication or initial screen to determine whether or not whatever therapeutic you developed is going to be functional.
Thanks, Anthony. Thanks a lot. Maybe Stefan, if you can please add on.
As said, maybe I can add some additional aspects. So of course, I would also differentiate between CARs and TCRs. So in respect to CARs, what people normally do is if you have already a very nice antibody which works in the clinics, of course, I would in a first trial use this one and clone it and try to make a CAR from a therapeutic antibody. On the other hand, we already know if you start with screening for antibodies that the highest binders, the highest affinity is not always the best for the CAR T cell. We face the problem that we have tonic signaling, that the CAR T cells really die off. So we need another measure to find which CAR would work best with a cell in a functional assay. As Anthony said, I think the functional assay is pretty important. A colleague of mine is screening actually CAR libraries in reporter cell lines, TrueCut reporter cell lines, and by this trying to identify with a specific antigen those which are best suited. So he can, of course, deplete first tonic binders, and then he can look which one is fastest and stronger. So this this functional assay is, I think, very important, and same for the TCRs. So what people do is they look for clone frequencies and select the highest clones, the most frequent clones. Okay, you still don't know whether this is against a viral epitope or this is specific. And we also try to define signatures, which could be used for identifying, but the approach I presented is agnostic to the antigen, and is also functional, because we see that these T cells secrete cytokines and work. Of course, we still have to do a lot of validation afterwards. So we need all the different techniques to get a complete picture before we have then something which can be applied.
Yeah, well said. Maybe another question, and it's on the same line. It's how do real-time biological readouts and multi-model data integration enhances our understanding of cancer immunotherapy efficacy? And Stefan, maybe you can start with that.
Yeah, well, what I like with the lightning experiment is that we see several cytokines at the same time, and can try to make an assumption on the kind of response of the cell, also whether 4-1BB is upregulated or not. This may be very informative, though as I said in my presentation, we are not yet at the point to have a full interpretation of these signals. Still, we have another measure of selecting TCRs, because we can go for speech, which is another parameter, and also for multimodal reactivity. I would assume that those are the better T cells. Still, the question in terms of the T cell receptor is, if I put that in another cell, I will have probably another response, kind of response. Maybe this is more a question Anthony can answer, add on the functional validation, because then it is really important to see how do these cells react with the new TCR.
Let's hear from Anthony.
Yeah, so I definitely think the real-time biology is critically important. I mean, one of the most interesting findings that we saw in some of our early CAR T cell studies was just that there's a lot of heterogeneity in the response, right? There's a lot of underlying biology that drives different functional responses, and there's different parameters that you can actually use to then start predicting whether or not a CAR T cell or a TCR T cell is going to be functional, once it's infused into a patient. And so one of the things we've been looking at is rate of cytotoxicity, which is something that, in real time, if you're imaging, the target cell and the effector cell, you can gauge that interaction between the two. And then how quickly after immune synapse is formed, does the CAR T cell kill the target, or does the TCR T cell kill the target? It actually gives you a little bit more indication too. There's, scenarios where you can actually see activation induced cell death, where, you would presume in more traditional assays that, whatever the readout is, that there's killing overall, but this gives you the, ability to actually distinguish between effectors and targets, right? So is it that the CAR T cell killed the target, or is it that the CAR T cell was overactivated and actually induced its own cell death? So I think that's an important parameter, but also, parameters like motility, right? Like how functional and how capable is that cell migrating within the pen, right? There's been a lot of interest, in the cell-based therapy field more recently, suggesting that, motility is an important parameter, because obviously once a cell is infused into a patient, it still has the goal of, reaching the target, interacting with the tumor, and then eliminating it. So I think, being able to, characterize those in real time is extremely important, and being able to look at multiple parameters obviously gives you better predictive value for, future studies, and then better correlations with clinical studies as well.
Yeah, and that's where the Beacon platform really shines. And Anthony, there is actually one question directly related to your presentation, so I'm going to ask directly to you. So in your presentation, you shared recording of time between T cell and target cell contact and killing. So is this done by Beacon platform, or did you record that manually?
Yeah, no. So one of the parameters, so we image every 30 minutes, while we're performing the assays. And so that's actually recorded every 30 minutes. And then we can actually determine, using these montage images, like how long is that interaction taking place? And so, yeah, those images were collected on the beacon, and then we could determine how quickly they engaged, and then, how long were they engaged with their specific targets.
Okay.
Actually,
Thanks, Anthony, for
that was my question. So you write down the timing and then calculate the time, or is that done by the
time
analysis
is
software?
yeah, that's part of the software. It analyzes. So on one of the CSV files, it actually gives you the time for when those images are captured. And then you
Great.
can generate those montage images, and at the bottom they actually have the time as well. And so you can actually monitor we perform the assays typically for 24 hours. And so you collect those images, in 30-minute intervals over a 24-hour period, and then you could determine that interaction.
I'm gonna try that. Was a nice paper actually, which you published on that whole subject. Enjoyed.
Yeah.
Thanks.
Congratulations, Anthony, for that. Yeah. And I think that's another beauty of the Beacon platform, enabling all these assays in a real time in a dynamic measurement instead of the endpoint assays, which really become very useful for cancer immunotherapy. Now, let's move on. I'd like to now ask questions for antibody discovery part, and these are for François and Xiaoxiao. And the first question for that is, given the limitations of classical immunization strategies and subsequent hybridoma generation, how does single-cell screening revolutionize antibody discovery, particularly for challenging targets? And maybe, François, I'll ask you to start the discussion.
Yes, I'm sorry, but I missed the end of the question. I'm sorry.
I'll restate the whole question. So given the limitations of immunization strategies and subsequent hybridoma generation, how does single cell screening revolutionize this whole therapeutic antibody discovery part? And particularly here, we are talking about challenging targets, which targets are hard to find.
Yes. So maybe I can start. So in a way, even for non-challenging targets, you really need to start with a collection of antibodies with a reasonable diversity and functional properties. So it's relatively easy without single cell to have such collection with classical immunization or even phage display or other techniques. But when it comes to a challenging target with a relatively narrow epitope, then you have to really go in depth into the repertoire of the B cell responding to the antigen, and in that case, only having a quick answer on many different candidates will give you the opportunity to select for production the best ones. Otherwise, you will produce hundreds of different monoclonal antibodies without knowing really if they are interesting or not, and you will lose quite a lot of time there. So really having the opportunity to have serial screening with different parameters of binding, but also blocking sometimes, cross-reactivity and so on, really can give you the opportunity to start with a collection of antibody without spending tremendous time to produce hundreds of antibody at the beginning.
Thanks a lot. So it's not only just a hit generation, doing some functional assays, bringing those lead optimization functional assays upfront, and doing those functional assays to get more functionally validated targets is helpful. So maybe, Xiaoxiao, I'll ask you to add on what François has just shared with us.
Okay, thank you. And yeah, so besides the reason that Dr. Frank just said, I think there are also some other reasons or other advantages for with the single cell system, especially Beacon. One thing is that I think because Beacon can let us to screening the plasma cells or the B cells directly without any amplifications or other modification step, which means there will be no biased step introduced before we start to do the screening. So I think that is very important to let us to pick up the rare sequences. So because for some difficult targets, we may need to focusing on some rare sequence that with very low frequency to show out, and which maybe that is the one we just looking for. And with other methods such as the phage display or even some other NGS based methods or the hybridoma, we always need to do some pre-treatment with our cells, all the sequences like amplifications, like diffusions. And for that ones, we always will get into the BIOS, which means we will lose parts of our diversities during this process. And that make us to just lost what we want or what we're looking into. So that's the one advantage I want to mention about Beacon. And the other thing is that by using Beacon, because we will do the sequential, although we do these different assays one by one on the machine, but actually we just do them in one round. And after that, then we will decide it, like if we do like 4 or 5 assays in one Beacon-based selections, we can always do the assay first, then we go back and check all the results and decide which assay is the most important criteria or which one is less important. So which means we won't do a funnel-based screening. We don't need to get rid of part of our positive clones by each step of the screening. We can always go back and review all the results and pick up what we want most. So that also will help us to getting what we real or desired from the Beacon screening. So I think these two are very important for when we're doing the screening or getting the antibody discovery with the very difficult targets. Thank you.
Yeah, thanks a lot. It's always a pleasure to hear from our customers about Beacon and how it's advancing antibody discovery. I do have another question on the same topic, and this is we all know that AI, artificial intelligence, is becoming more prominent for antibody discovery field. So a question came in, what is your view on the impact of AI and ML on therapeutic antibody discovery field? And how do they complement your usage of the Beacon system? And Francisco, I'll ask you to start again.
Yes. So I must say we are not using AI at the moment, so I'm maybe not the right one to answer this question. But I saw that Sun Chen does it, so maybe she can have a better
Absolutely.
answer for me.
Let's do that. Let's go to Xiaoxiao.
Okay, thank you. So for that one, actually, we are currently not really combine the AI or the machine learning together with Beacon yet. We just comparing the two different kind of screening method, and so currently I think AI is really important, or we're getting more and more important for the antibody discovery in the future. But currently, I think the method we already have still cannot really help us to pick up the right antibody among all different sequences or just based on sequences informations. So if we comparing the Beacon and AI directly, then I definitely will choose Beacon against AI because it will be much more efficient, especially if we want to pick up the antibodies that need to to match a lot of different binding or functions or different criteria. But if in the future, when the AI's method getting more and more mature, I think that will definitely help us because after Beacon screening, especially when we're using like more than one chip and with a higher throughput, we may getting a lot of sequences after that, hundreds of them. So during this time, what we do is we will analyze the sequences and doing the, like traditional clustering and checking for the PTMs, and then pick up the representative sequences based on these informations. And when we go back to check all these things, we found that sometimes we may really lose some of our good binders because we cannot really pick up the right representers from each clusters. But if the AI really works, it will help us a lot when we help us to just reviewing all the hundreds of leads we already get from the beacon, and help us to identify some other properties like the biophysical properties from the sequences, or maybe some other, like I check in all the bindings, and probably the AI will help us to know the epitopes from the sequences. If it's really mature, then that I think will really help a lot combined with beacon.
Still a long way to go to predict based on the sequence, but no, I just want. So I completely agree with that. What we do right now is, as we have sometimes many sequences, even without AI, we can modelize the antibodies because there are software available for that, and make scores according to PTM, so liability data that are well known, and we can also have a score of the stability of the antibody based on the hydrophobic content in the CDR, for example. So even without AI, the fact to have the sequences before and the clustering, of course, of those sequences, you can really, based on all what is known in the antibody field and the modelization of the antibody already available, quite a good selection of and discards the obviously non-developable antibodies, for example. I also agree that doing that is also sometimes you lose some very interesting binders, but there is always a lot to do based on the sequences and the available modelization to select for the most probable developable antibodies.
Yeah, that's true. Yeah, thank you.
Yeah. Okay. Thanks a lot. Maybe we'll move on to our third application. I do want to ask Dr. Shu Wang, some questions came in for cell line development. And the first one is, how does the Beacon platform improve the efficiency and accuracy of cell line development for biopharmaceutical production, especially for complex products like bispecific antibodies?
Hi, so can you hear me?
Yes.
Yeah, okay. So thank you for the question. So we have been using the Beacon platform to produce monoclonal antibodies. It's very successful, but we also have a bispecific antibody to produce. Before, we don't do the quantity assay, just like the ultimate binding assay I talked about. So, and turn out, so we have many high titer clones in the end, because the quantity not good, so we have to toss them away. So if we can use a optofluidic system, the Beacon system, we can do a ligand assay together with Spotlight HUFC. We can measure the concentration of FC and other single arms. We can calculate the ratio. Then we can select good clones that will improve our efficiency of cell line development, and we can evaluate more good quality clones, and we can have good tighter clones also. Yeah, I think it's very helpful.
Thanks a lot. Maybe one last question from our audience today. This is again for cell line development. What are the critical parameters and techniques involved in accurately measuring heterodimers and optimizing clone selection? And again, for you, Shu.
For the method to measure the heterodimers, homodimers, halfmers accurately, that's very critical. For our bispecific antibody, we use a charge, different charge, negative charge, a positive charge to form heterodimers. So we use the CX HPLC to measure, percentage of different heterodimer, homodimer, or halfmer. But sometimes it's different. It's difficult. So because there's a deamidation and amidation, it will change the charge. It's hard to differentiate, heterodimer from the homodimer or halfmers. So recently, our scientists developed a new technology. It combines CX HPLC with mass spec to differentiate and measure the percentage of different homodimer, heterodimers. It works very well. It's better than CX alone because you know which one is which, and you can also calculate the percentage.
Mm-hmm. Thanks. Thanks a lot. I do have one last question. This is question from me, and this is for the whole panel. And the question is why did you select the Beacon platform or Lightning platform, and how did it facilitated your research in your lab? And let's start from Anthony, and then we'll go through the whole panel.
Yeah, so I always like to share the story. When I was a postdoc, I was working on trying to develop TCR T-cell based therapies, and really the pipeline to discovery that we were utilizing started at, the discovery, went through validation, and then ended with translation. And, we wanted to improve the resolution of the assays we're using. So we obviously wanted to go to single cell. One of the challenges we face is, a lot of this, single cell platforms at the time, they give you a one parameter readout. So you could look at the transcriptome, you could look at, cytokine secretion, and then in a separate assay, you could look at the phenotype. But, you could never link all of those parameters, back to a single cell. And so it was challenging to know whether or not, that cell that had the most cytokine secretion was also going to be the best killer, and what the phenotype or the transcriptome of that specific cell was. And so for me, I was looking for a platform that would allow me to link all these parameters, because obviously, the more data that you can, collect on a single cell, the more informed you're going to be when you're starting to make decisions, especially when you're trying to translate a therapeutic. And so it was really that, being able to link everything to a single cell, looking at multiple parameters, and then also, just the throughput. So, being able to characterize multiple cell types, being able to compare different, TCR T-cell constructs. And so really just the flexibility in the number of readouts that you could perform, and the fact that it was at single cell resolution, to me, was very enticing.
Yep. Well said. Stefan, if I may ask you to go next.
Yeah, for me, the interesting question was, there is a platform where I can go looking for specific T cells without knowing the antigen, so being agnostic to the antigen, and start with the function. In most other assays, you start with the antigen or you start with a mutation, and then you search for T cells which recognize this neoepitope, which is then difficult. You don't know whether this is really expressed on the target cells, and a lot of other questions. Of course, this approach also has its disadvantages because, as discussed in my presentation, probably the authorities want to know what is the antigen, so we need to find it out anyway. But we start with a functional already, with a cell where we already know that it functions in its recognition against the target cell. And we did not use the caspase assay. Actually, this allows even killing. So we were looking only for recognition. But I think there are so many different options here which this platform can be used. I like this.
Okay, wonderful, awesome. And Xiaoxiao, maybe you go next.
Yes. So I think for the beacon for the antibody discovery in Nona, one most important thing is really quick, because we have our own humanized mice. So for immunization, it's not really a big deal for us. But after immunizations, by traditional method, we need to take in months to screening one target, but using beacon, yeah, it's just a 1 day to the screening and plus the later sequencing and 24-well confirmation for the recombinant. It's just within 1 month, and make everything very clear. And another thing is that because the beacon can help us to increase the throughput for the screening, so it will be much easier for us to screening those very difficult targets, because we can start from more clones, and to just getting those clones has less than 0.1%, even much lower, the percentage to get very with low frequencies, and we can still find it out. So currently we are not really, before that, when we're doing the immunization and we're checking for the test bleed titer, and we will see like if we want antibodies that can recognize target A but no target B, no target C, and again cross with target D, that will be really, really painful work, because when you're checking the titer, you can never find a mice that show the best titer as you desire. But currently we know that even we pick up a mice show the average titer against A, B, C, D. But when we run the assay on beacon, we can still find the exact cells that can just meet our criteria that recognize A and D, but no B and C. So that will definitely help a lot. Thank you.
Yeah. Okay, François.
Yes.
I guess.
Well, it was quite an investment for us to change technologies, and so we tried to see several types of solutions there before engaging with the beacon system. What really I wanted is like Shushan to be quick and to go to the single cell level, and there was alternative technologies such as cell sorting by flow cytometry, for example. But really what I wanted was a technology that could allow a selection based on the native antigen. So, for example, you cannot sort B cells with a flow cytometer if you don't have a recombinant antigen. But usually for difficult antigen, you don't have that. You don't have a good recombinant antigen to fish the cells through flow cytometry experiments. So that's really what made the difference. The fact that we can use transfected cells, so we know that's a native antigen that you screen on. And that was, in fact, the only platform that allowed that at the single cell level, with the advantage, of course, of the speed and the single cell analysis with the depth of screening. So that's what was making the difference. And now you can really interrogate also different organs, because when you have an immunization, as Shushan said, it's not always at the same place. So you can interrogate lymph nodes, you can interrogate bone marrow, you can interrogate spleen, and all that native antigen, even if the antigen is very difficult, because you can use transfected cells.
Yeah, well said. Okay, last but not least, Dr. Shu Wang, I'd like to hear your thoughts.
Okay, so we use beacon for cell line development. So I think beacon is very powerful and efficient. before we use the beacon, we use FACS or limiting dilution or cell plating to load single cells to wells many plates. The after cells are confluent. We will do a screen normally with the help of automation. Because we have to screen 60 to 100 plates. We will screen a supernatant with octet or ELISA. So after we get the top clone, 96 or 48 clones, it depends on two different labs. Then we will export them to 96 well plate. For beacon, so one equipment can do all the equipment job. We can use the beacon to load cells to nanopads, and we can screen many clones. We can do the giga-assay earlier, then we can screen many clones, like 10,000 clones in 4 chips, and export the top clones. Very simple, and also we can screen clones earlier. And right now we are developing the all-chip binding assay. Later, maybe we can use this method to screen clones quantity, not just titer. Yeah, I like beacon.
Yeah, thanks. I love to hear that you like beacon. So that's wonderful. Okay, this brings us to the end of our roundtable discussion. I would like to thank all our participants and attendees for their time today. I'd like to especially thank Dr. Xiaoxiao Wang. She joined us from China. I know it's 3 a.m. in the morning. So thanks a lot for joining us live for this roundtable discussion. Thank you. And this will conclude our presentation in auditorium. However, our booth and poster hall will remain open for next 1 hour. Feel free to interact with our technical experts live and ask any question you have, or discuss any potential collaboration projects. So I'll highly encourage you to go and visit our booth and as well as poster hall. Thanks, everyone.
Thank you.
Thanks a lot.